Mr. McGowan here, and today I'm going to show you a typical question that we do in grade 12 genetics, and that is to figure out what the mRNA transcript would be from a DNA sequence, and then to use translation and change that mRNA transcript into a sequence of amino acids that would happen at the ribosome. So just to remind you that with the genetic code, which you see down here, they are based on a triplet of nucleotides. So nucleotides are placed in sequences of 3, and that's the genetic code. To use the genetic code, we only use the messenger RNA. There is a genetic code for DNA, but that's not what this table is based on. This table is based on messenger RNA. So the first thing that I like to do is to divide up the DNA into triplets. So I'm just going to put a little slash line every 3 just to indicate where those triplets would be. This is only for my organization. It has nothing to do with the process that really happens in the cell. The next step that would happen inside the cell, though, in the nucleus during transcription, the RNA polymerase would join on at the beginning sequence. So the beginning sequence is called the promoter, and the promoter knows where the beginning of the gene is because of a start sequence. The start sequence is always going to be TAC when it comes to the DNA. And what does that code for? Well, remember, T corresponds with A. A, because this is RNA, corresponds with U. Then we have G. Then we have U and a U and a C. U and an A and another U. Let me just put little slash marks between them. There we go. Then we have a C and a G and an A U, a U and an A G, G and an A. We have a U, a G, and an A. We have a C and an A and another C. Cat, how fun. We have a G, we have a U, and we have an A, and at the end we have a U and an A. Now there are no codons which have 2 nucleotides, so that wouldn't count for anything. So now we have our codons. The next thing is to use the genetic code down here to actually convert them into amino acids. So this would be part of translation. Translation happens in the cytoplasm when the messenger RNA joins on to a ribosome. A ribosome actually reads the codons on the messenger RNA. A tRNA would then come in and place down the appropriate amino acid according to that code. So to use the code again, what you need to do is read the first letter on this side, the left side, second letter on the top, and the third letter along the right side. First base, second base, third base. You didn't even know we were playing baseball. So every protein sequence will always start with methionine, AUG. You may remember that from the notes. So AUG, A, U, and then you go down to here to G, AUG, methionine. This particular genetic code only has abbreviations for the amino acids. I will say them you don't need to know the abbreviations. I also like to put numbers. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's just, again, for organization. Next we have UUC, U, U, C, phenylalanine, P-H-E. Third one, UAU, U-A-U, T-Y-R. That stands for tyrosine. Fourth one is CGA, C-G-A, arginine, A-R-G. Number 5, UUA, UUA, leucine, L-E-U. Sixth one, GGA, G-G-A, glycine, G-L-Y. And we're on 1, 2, 3, 4, 5, 6. Number 7, UGA, UGA. Oh, that's a stop codon right there. Look at that. So that means at the ribosome, it would indicate termination. The ribosome and the messenger RNA would actually separate, and that would be the end of the polypeptide sequence. Well, what about these ones at the end? They wouldn't actually be coded for at all. They might exist on the mRNA, but they wouldn't actually code for any amino acids in the sequence. Just for fun, let's just find out what they are. CAC, CAC would be histine. GUA, GUA would be valine. And this little UA, it's not a codon. It's just garbage, basically. It doesn't code for anything. Hopefully that helps. I will be putting this file on as well for you to practice. If you have any questions, don't hesitate at all to email me. Thank you.